My name is Cesar from Heather. Today I am going to show you how to use the string database, which is a very excellent database for the protein interaction analysis and the system biology related analysis. मैं आज बता रहा हूँ आपको बताऊंगा कि स्ट्रिंग डेटाबेस को किस तरह इस्तेमाल करना है और ये प्रोटीन इंटरेक्शन और सिस्टम बायोलॉजी में किस तरह वर्क करेगा. सो लेट्स स्टार्ट विद clicking on the first sites coming after putting the keyword named as the string database. स्ट्रिंग डेटाबेस आपने सर्च करना है गूगल क्रोम के अंदर और आपको सामने जो पहली वेबसाइट उसको क्लिक कर देना. दिस इज द वेबसाइट दैट कम द फर्स्ट and after clicking the link of the website you will get such interface. जब आप पहली वेबसाइट को क्लिक करेंगे तो इस तरह का आपको इंटरफेस नजर आएगा. सो सबसे पहले आपको ये बताता चलूँ कि सिस्टम बायोलॉजी क्या होता है कि हर जानदार के अंदर जो कुछ एक सिस्टम होता है, इंटरकनेक्टेड प्रोटीन्स और जीन्स के एक सिस्टम होता है जो आपस में जुड़ता है और जुड़ने की वजह से वो पाथवेज के अंदर बहुत ज़्यादा प्रोटीन्स और डीएनए वगैरह होता है, प्रोटीन्स और जीन्स वगैरह होते हैं, वो इंटरकनेक्टिवली वो एक पूरे पाथवे पूरे फंक्शन को वो काम करवाते हैं. जिस तरह फॉर एग्जांपल क्रैब साइकिल हो गया और इस तरह की हज़ारों एग्जांपल्स हो गया, एंजियोजेनेसिस हो गया उसके अंदर ऑन्कोलॉजी के अंदर. दिस सिस्टम बायोलॉजी इज इंटरकनेक्टेड सिस्टम that exist in the living organism and in the pathway or in the system biology the in system biology the pathway exists. What are the pathways are the interconnective frameworks of the proteins and the genes when they work in interconnectively and they perform a specific function. बहुत जगह यहाँ बहुत ज़्यादा ये होता है कि जब हम रिसर्च कर रहे होते हैं तो हमें क्या करना होता है कि हमें ये देखना होता है कि कौन से जीन्स कौन से प्रोटीन्स आपस में इंटरेक्ट कर रहे हैं और उससे हमने कोई एक अजम्पशंस क्रिएट करनी होती है. रिसर्च पेपर के अंदर हमने कोई अजम्पशन या कोई हाइपोथेसिस देनी होती है या हमने कोई एक फैक्ट को वैलिडेट करना होता है. तो अब हम हमारे पास इतने इक्विपमेंट्स नहीं होते कि हम लैब के अंदर उसके डायरेक्ट फिजिकल लिंक देख सकें कि वो एक जीन दूसरे जीन्स के साथ नेटवर्क बना रहा है, इंटरेक्ट कर रहा है या नहीं कर रहा है. तो साइंसदानों ने अपनी आसानी के लिए जितने भी एक्सपेरिमेंटल वैलिडेशंस किए हुए हैं कि कौन सा प्रोटीन किसके साथ वो जुड़ता है, जितने भी एक्सपेरिमेंटल प्रोसीजर्स किए हैं उनको एक डेटाबेस एक इंफॉर्मेशन की फॉर्म में रख दिया गया है. तो वो रख दिया गया और आम आवाम के लिए वो खोल दिया गया. So if we want to see the protein interaction between the if we want to see the protein interaction between the proteins, so uh, so uh, actually everyone cannot afford the labs. For example, if one is seeing the interaction of the one protein with the other protein, he or she have to go to the lab, and then he have to check the physical interaction by the very expensive biological experiments. Okay, so this is very time expensive and time consuming thing. So to avoid these things, all the experimental validated protein physical interactions have been reported in a database known as the string database. So simply, for if you want to see the uh, one gene in one gene in one organism have the how much proteins are interacted with that protein. Okay, so you have to put the protein name in the protein name section and check the organism and select the organism. By clicking at this, you can select the organism. After entering the name of the protein and entering the name of the organism, okay, for example, I want to enter the name of the Homo sapien. You have to click here, and after clicking you have to wait some time. अगर आपने कोई प्रोटीन का नेम डालना है, चेक करना है कि एक प्रोटीन एक जानदार के अंदर कितनी और ज़्यादा प्रोटीन से स्टैटिस्टिकली सिग्निफिकेंट वो रिलेटेड है, उससे उसके साथ जुड़ती हैं, तो आपको क्या करना है, नाम डालना पड़ेगा और ऑर्गेनिज्म सेलेक्ट करना पड़ेगा. फॉर एग्जांपल, आई हैव टू सेलेक्ट द होमो सैपियन, देन आई क्लिक ऑन द सिलेक्ट एंड देन क्लिक ऑन द सर्च. मुझे क्या करना पड़ेगा, नाम को सेलेक्ट करने के बाद उसको क्लिक करना पड़ेगा। उसके बाद आफ्टर दैट you have to once read the first protein TP53 is the TP53 is the gene name and P53 is the protein name. So it is the same one protein that I am going to research. So when I have if I select this, then it will be wrong, it is the another protein. So you have to select the relevant protein. I already know the function of that protein, so it is the same protein. Then you have to click on the continue. When I have clicked on the option continue, a such graph will appear, a such figure will appear. At looking the first, it looks like a very weird thing, but it is a very interesting thing. Uh, so these things are actually are the protein or the gene names. Okay, and these things are known as the nodes. These are called nodes. ये जितनी भी छोटी छोटी गोल गोल सी चीज़ें हैं, इनको हम क्या कहते हैं, इनको हम नोड्स कहते हैं। और ये जो धागे से ये जो लाइंस हैं, these lines are called as the edges. Okay, these are nodes, these are edges. So क्या होता है कि ये TP53 प्रोटीन हमने इंटर किया था, उसको इंटर करने के बाद ये कुछ और 7 प्रोटीन आ गए, और वो प्रोटीन TP53 के साथ धागों के साथ जुड़ी हुई है। What it means, when we hit the continue, we will get the result like this. Okay, so what it means, uh, this uh, this node is connected with all other nodes through the small strings. These strings are known as the edges, and the other strings name known as the nodes. Okay, so what it represents, ये क्या जाहिर करता है, ये जाहिर करता है कि यहाँ पे फॉर एग्जांपल पांच छोटे छोटे धागे जुड़े हुए हैं RPA1 के साथ। इसका मतलब है ये जो TP53 है, ये बहुत ज़्यादा, जितने ज़्यादा धागे से होंगे, जितने ज़्यादा स्ट्रिंग होंगे, इसका मतलब ये है कि ये TP53 की इसके साथ जो एसोसिएशन है वो बहुत ज़्यादा क्या है, वो बहुत ज़्यादा गहरी है। उसको फॉर एग्जांपल यहाँ धागे से कम है, इसका मतलब है इसके साथ एसोसिएशन कम है। फॉर एग्जांपल if we look at the nodes and see the numbers of the edges, for example here the number of edges are more and here the number of edges are less. So we can so it is the assumption, and we cannot show the assumption, we can't say the assumption, it is the fact that here are the association between this and this protein is much more as compared to this and this. Okay, so we can see the interaction of other proteins like this. If we have a single protein, this is also called a hub protein. In our case it will called as a hub protein. हमारे केस के अंदर ये hub protein कहलाएगी। और स्ट्रिंग डेटाबेस के अंदर हम एक प्रोटीन, सिंगल प्रोटीन से होमो सैपियन के अंदर हमने देख लिया कि वो किस तरह दूसरी स्टैटिस्टिकली सिग्निफिकेंट वो कौन कौन सी प्रोटीन है जिसके साथ ये TP53 बहुत अच्छे तरीके से जुड़ता है। तो जब व्हेन वी क्लिक ऑन दी एनालिसिस, when we click there are the many options appear here. When we click on the analysis, we will get the results like this. So this section is very important. When we click on the analysis, their number of node, there will be written as the number of node 11, means when we enter the protein, the database selected the search through the algorithms and found that there are 11 edges, 11 nodes are present, and number of edges like these strings, and the total number of edges that are interconnectively with the other proteins are the 35. And the very interesting thing is the expected number of the edges. The database expected that there should be at least 26 number of edges. If the number exceeds from this number, then the results are good. Okay, the TP53 results, the TP53 interactions are good. If the results if the number of edges or less than number of edges of the expected number of edges, then the interaction is not good interaction. Okay, when we do a multiple protein analysis, then I will tell this thing in detail. Okay, so there is also option of average and not degree. What it means every every node has the interaction with the other node with an average score of 6.36. It means there the average the average score of the strings with the other strings is the 6.36. Okay, this is the average. Okay, not a whole. So this stat is very important, and this gives the a very good insight of our analysis. The PPI, protein enrichment value, it means that the p value of our scenario is 0.0582. As you are a researcher and you know the value of 0.05, and I think I should not explain it. Okay, so you have to go to the export. So you can also give the symmetry like this by making the different design. You can also make a publication ready figure by making a different design. For example, you can do this. Okay, and what is your wish, you can do. Okay, so after doing this, you have to click on the option download. Okay, when you have clicked the option download, very high quality picture will appear. Okay, this is a picture. Okay, this is not a black picture, it is a background free picture. You just have to click on the copy and you have to put in your research paper. Okay, this the other thing is the viewers. So there are if I want to see that the what are the experiments are conducted टू वैलिडेट, अगर हम ये देखना चाहते हैं कि वो कौन कौन से एक्सपेरिमेंट्स किए गए हैं जिसमें ये सारे प्रोटीन इंटरेक्शन एनालिसिस को उन्होंने वैलिडेट किया गया है. सो यू हैव टू क्लिक ऑन द एक्सपेरिमेंट्स. if you want to see the what physical experiments have done to see the protein interaction of TP53 with the other proteins, you have to click on the experiments. By clicking on the experiments, you can see protein interaction directed by the anti-tag co-amino precipitation assay. It is a molecular or the amino chemistry related assay where the uh, where it is found the interactions with the TP53. The all proteins mentioned here are validated with a medium confidence score, average detection medium confidence score, that TP53 showed the physical interaction with the other all mentioned protein. So the question is why there only 11 globular structure appears. It is appears on the basis of statistical scores. So for example, if we it is shown here, the TP53 also show a link with the for example ACTR3. When we click on ACR3, and the here whole description will appears, and you can also see the structures and the motifs and the other things. Okay, so so the TP53 interaction with the this gene is not too much statistically important. Okay, so that is why it is not appeared there. So we can see the interaction on by clicking on the experimental experiments, and we can also click on the database. What is the functioning of database? Let's see. So we can see the database knowledge here. So database knowledge here gives us the relevant data sets in the human Homo sapien. For example, the annotated KEGG pathway shows that this gene has the important function in the cell cycle, and you can also see the KAGG interpretations uh, in the other pathways. Okay, this is related to the pathways or databases, metabolic pathways and protein complexes, similar transduction pathways. Okay, so this is the related the pathways, and when I click on the text mining, if I want to see that which all the related text about the TP53, okay, and their interactions, related pathway, the this database fetches the information from the PubMed and the other sources. अगर हम ये देखना चाहते हैं कि TP53 का PubMed के अंदर कौन कौन सा डाटा अवेलेबल है इंट्रोडक्शन के रिलेटेड, तो हम क्या करेंगे कि इसको क्लिक करेंगे, टेक्स्ट माइनिंग को क्लिक करेंगे, तो हमारे पास सामने ये टेक्स्ट आ जाएगा. हमें अकेले अकेले पढ़ने की जरूरत, वी डोंट नीड टू रीड वन बाय वन, जस्ट वी हैव टू क्लिक ऑन द समराइज ए. By clicking on the summarize AI, you will get the summary of the AI based summary of the TP53 interactions and the other related content. Okay, so just PubMed ID and all the other thing. It is the role of the TP53 in the family in cancer. Okay, the major role is the TP53 in the cancer. It is a very well known role, and the thousand of studies have been conducted. Okay, so this is the text mining is a tool in the string database that is used to see the information about the relevant protein. Okay, you can also see the fusion, co-occurrence, co-expression, genes that are sometimes fused into the single operating frame. Sometimes it happens that if one protein, if one gene is fused with the second gene, when the translation when the transcription occurs, the protein the protein is translated with the fused, the two genes that are fused are translated into a whole one protein. Okay, until the stop codons occur. Okay, you can also see the information about the gene fusions in the different organisms. Genes families whose occurrence patterns across the genome show the similarities. You can also see the co-expression. These are the advanced concepts, and I will conduct a lecture on all things in very details. So I hope you will grasp a very good concepts about the single protein by name identifier. For example, if you have a multiple proteins and you want to see the interaction about that proteins, you just have to in many scenarios in research, you always use the multiple protein option. If you have multiple proteins, for example, if you did the transcriptomic analysis and the other related analysis, you just have a list of the proteins. Okay, so if you want to see the connections about a list of proteins, so you have to go to the multiple proteins and just put the multiple proteins. Okay, I put the multiple proteins. Okay, you can also put the multiple proteins in a txt file, and you can browse from your hardware and select your organism. For example, I want to see the protein interaction of all the proteins that I have generated during transcriptomic analysis and any type of analysis. Then I can select the organism as a human Homo sapien, and I can sort I can conduct on search, click on search. अगर मैंने ये देखना है कि मल्टीपल प्रोटीन्स मेरे पास हैं, ट्रांसक्रिप्टोमिक एनालिसिस में इस तरह होता है कि अगर बहुत ज़्यादा हमारे पास प्रोटीन्स होती है और हमें देखना है कि आपस में उन प्रोटीन्स का क्या लिंक बन रहा है, क्या वो स्टैटिस्टिकली सिग्निफिकेंट आपस में मिल भी रही हैं या नहीं मिल रही है, तो मुझे क्या करना पड़ेगा कि मुझे उनको सारी प्रोटीन्स को उठा के उसके अंदर स्टैंडर्ड उनकी जो एब्रीविएशन, उनका जो नेम होगा प्रोटीन या जी नेम उसको वो स्टैंडर्ड होना चाहिए, उसको मैंने डाल देना है। 50 प्रोटीन थी मेरे पास, सबको 1 बाय 1 चेक करना है कि वही प्रोटीन है उनके फंक्शन से, ठीक है ना, इवन के इतना चेक करने की ज़रूरत नहीं होती। So you just have to check all the proteins that they have the same functions, and if when you have validated and you have checked the protein, you just have to click on the continue. The same like the in single analysis. As such a figure will appear. Uh, these figures show a very dense and very good and very statistical and statistical significant interaction. I showed this by seeing the too many uh, strings. It is still loading. लुक, ये देखिए कितनी ज़्यादा यहाँ पे स्ट्रिंग्स बन रही हैं, कितनी ज़्यादा स्ट्रिंग्स बन रही हैं। मैंने पहले आपको समझाया कि जितनी ज़्यादा स्ट्रिंग्स होंगी इसका मतलब है कि प्रोटीन प्रोटीन इंटरेक्शन उतना बहुत ज़्यादा गहरा होगा। और ये हमारे पेपर के लिए, अगर हम इस तरह का हमारी स्टडी है जिसमें हम प्रोटीन प्रोटीन इंटरेक्शन दिखाना चाह रहे हैं या यही शो करना चाह रहे हैं कि इसका आपस में तल्लक है, जितना ज़्यादा ये गहरा होगा, जितनी ज़्यादा लाइन्स होंगी उतना अच्छा ये हमारा पेपर होगा, उतना अच्छा हमारे लिए ये एनालिसिस होगा, ठीक है। अगर हमारी सिर्फ एक लाइन या कोई भी लाइन ही एक अलहदा आ रही है तो वो नहीं होगा। तो ये देखें कि ये प्रोटीन्स आपस में कितनी ज़्यादा अटैच हैं। you can't imagine you can't imagine you can't you can't see, you can't see you can't expect the you can't count these strings. So you have again go to the analysis and you will see the score will be very high. So expected number of edges 352, but number of edges existed in the whole in this figure is the 929। तो यहाँ पे क्या हो, एक्सपेक्टेड नंबर ऑफ एजेस क्या होने चाहिए थे सॉफ्टवेयर के मुताबिक 352 होने चाहिए थे, लेकिन यहाँ पे कितने हैं, वो 929 है। कितना ज़्यादा डिफरेंस है, यानी कि इतना ज़्यादा ये नंबर ऑफ एजेस हैं, मींस कि ये इतना ही ज़्यादा अच्छा एनालिसिस है। ये हमें कहाँ से पता लगेगा, पीपीआई एनरिचमेंट पी वैल्यू से 1.02 रेज़ टू पावर -16 हमने ये पता है कि अगर 0.05 हो पी वैल्यू तो वो बहुत कम होती है। अगर 1.0 की वैल्यू -16 कर दी जाए तो वो कितनी स्मॉल वैल्यू होगी, मींस के ये जो एनालिसिस है ये बहुत ज़्यादा स्टैटिस्टिकली रिलायबल एनालिसिस है। So this analysis very statistical reliable analysis by seeing the value is 1.0 power to -16. Okay, so this is very good analysis and a very statistical analysis, statistically good analysis. So the viewer will not reject your this analysis. Okay, so hope so that the second viewer will can reject it, because the second reviewer is very brutal reviewer. So as you can also export the figure by clicking on the high resolution bitmap. Okay, so you can also use the other XML summary and the tubular output. You can use the some output in the Cytoscape and the other things I will show you later. यहाँ पे बहुत ज़्यादा फाइल्स होती हैं, जिस फाइल के अंदर भी, जिस फॉर्मेट के अंदर भी हमने डाउनलोड करना है, SVG TSV या किस पे भी करना है, तो हम यहाँ से कर सकते हैं। फॉर एग्जांपल फिलहाल तो मैंने PNG फॉर्मेट के अंदर करना है, क्योंकि मैंने पेपर के अंदर वो फिगर लगानी अभी डाउनलोड हो रही है। द साइज ऑफ द फिगर इज 5.4। It means that there are many edges and edges and nodes, so that is the size of the figure is very high. Okay, so once it downloads I will see you show you the. So here is a you can also change the color of node. Okay, you can also change the color of nodes. Here is the figure downloaded. Have a look. This is the high resolution figure, and all the edges and nodes have been shown. Okay, in the details of whole proteins here, and export clusters. You can also use you can also apply the clustering. Uh, these are some advanced concepts. I will tell you later. The video is going very long. Okay, so cluster, you can you can set the uh, scores by default by using your own parameters, or using your own values. Okay, settings, you can you can do your settings here. Okay, so the thing is the score, the medium confidence, there are there are some settings which can help us to robust our analysis. Okay, so you can, if we select the high confidence, the analysis will become very robust. Okay, the by default is the by default value is 0.400. Okay, so you can do this. And here is the option, enrichment display setting. Okay, the functional enrichment in your network. There is some error in the some backend error in the string database. So I want to show here a functional enrichment analysis. So it will be a very helpful for the for study the KEG pathway. Okay, so when it will when the website developers fix this issue, then I will show you the functional enrichment KEG pathway related analysis. Okay, so this is the whole video about the string analysis, data base. Okay, I hope you will understand this video and pray for me.